Hey everyone, welcome back to Windows Seed Studies. I hope you all are doing well. So we were discussing about cell biology, in which we in the last lecture we discussed about membrane proteins. Today we'll be discussing about the transport that occurs across the cell membrane. So today we'll be discussing about the types. Basically, we will be focusing about the types of the transport, and we'll also learn about the molecular mechanisms of those transport, energy dependencies, the structural features that the transport proteins have, and some physiological examples of those transport types. So let's get started with today's lecture. So we will be learning about transport across the membrane. So depending on whether the cell utilizes energy for bringing out the transport across the membrane, transport is basically divided into two main categories that you might already know from your high school level: the passive transport and the active transport. So the passive transport is the type of transport of the molecules where cell does not require any type of any form of energy. So it does not hydrolyze ATP, or it does not use the energy which is driven from any kind of concentration gradients. While in the active transport, cell needs energy for pumping out or pumping in certain molecules, and that is why active transport is the one where cell utilizes energy. It could be the direct use of energy or the indirect use of energy. So the passive transport, depending upon the which kind of molecule is being transported, whether it is a solid molecule or solvent molecule, or depending upon the requirements, the permeability requirements of membrane, it is categorized into different parts. So I'm going to simply define different types of the passive transport that are found in the cell. So the very for the first one, which is very known to everyone, it is the diffusion, simple diffusion. So diffusion is the type of transport where the small molecules, which are non-polar, they move directly through the phospholipid bilayer. Well, as we know that the membrane, plasma membrane, which is made up of lipid bilayer, so lipid is a amphipathic molecule, right? So it has hydrophobic part as well as hydrophilic part, and that is why only non-polar molecules are the ones which can directly which can directly cross the plasma membrane. So the non-polar molecules, which are usually small, they can directly, depending on the concentration gradient, they can directly cross the plasma membrane. So that type of transport is known as diffusion or simple diffusion. So it does not require help of any kind of protein or any kind of enzyme. It just follows the simple laws of physics. So wherever the concentration of molecule is high, the molecule is going to move from the higher concentration towards its lower concentration across the membrane without the help of any protein. So that type of transport is known as diffusion. So diffusion is shown by small non-polar molecules. Okay, well, large non-polar molecules, they could also show diffusion, but the rate of diffusions that vary. So any kind of non-polar molecule or any kind of uncharged polar molecule, it can cross plasma membrane, but the rate of diffusion or the extent of diffusion, it varies. But the charged polar molecules such as ions, they cannot pass the plasma membrane because, well, they have charge, so that is going to provide some difficulties for that molecule to pass. So ions, they cannot pass through the plasma membrane directly, so they need the help of certain protein to move according to concentration gradient, and that type of passive transport is known as facilitated diffusion. So we are still calling it diffusion because it is, it is occurring downhill. Means the molecule is moving from the higher concentration towards its lower concentration across the membrane. So it is a type of diffusion, but unlike the simple diffusion where you do not require any kind of protein or any type of help, the molecules, they are simply moving downhill. In the facilitated diffusion, the downhill movement is aided. Okay, and that is why it is known as facilitated diffusion. So you need a protein molecule to facilitate this type of diffusion, and that is why it is known as facilitated diffusion. So this is, the very famous example of this is aquaporins. So aquaporins are the channels which does the transport of water molecules. So depending on the types of the proteins that are going to carry out facilitated diffusion, there are two different types of proteins that could bring out this facilitated diffusion. The first ones are known as transporters, and the others are known as channels. So channels, so in last class, when we were learning about membrane proteins, we learned about different types of membrane proteins, such as transmembrane ones, the integral membrane proteins, which are, same, and the peripheral membrane proteins, the lipid anchor membrane proteins. So there are different proteins, the transmembrane proteins, which could form pore in the plasma membrane. They are known as the channel proteins. While there are certain proteins which, upon binding, upon the solid binding, they are the conformation of that protein changes, and now they can carry out, they can pass down that solid molecule from one side of membrane to the other side of membrane. So whenever the conformational changes occurs in the type of in the protein which is going to carry out the transport, that membrane proteins are usually known as transporters. So transporters, they have other names such as carriers, because they are literally carrying the ligands or the solid molecules which are going to bind to that protein, and they are also known as permeases. So you know that whenever we use ases, that is usually used for enzymes, because this is fixed in the biology. Whenever you see ase, flipases, flopases, lipases, so they are the enzymes which they are the category of enzymes, right? So why do we call transporters as the permeases? This is due to, because so permeases means they are going to promote the permeability in the membrane, because they are carrying out the transport from one side of membrane to other side of membrane. But why the permeases, which are a type of enzymes, they are also come in the category of transporters? Because transporters are the ones which have the mechanism, their mechanism similar to the enzymes. So the way enzymes get saturated upon the substrate binding, transporters, they also get saturated. Channels does not get saturated because they are literally pore forming membrane proteins. So let me draw a schematic for you. So let's say that this is a plasma membrane, okay, and let me just, okay. So in this plasma membrane, there's a protein, okay, which is directly forming a pore. So through this pore, the different types of molecules can slip in or slip out. Okay, so this type of transport which happens by the pore formation by the proteins in the plasma membrane, those are known as channel proteins. So the example is aquaporins, okay, which transport the water molecules across the membrane. But if there's a protein. Okay, this is pardon my schematic. So this is a transporter. Okay, so what happens is that whenever a certain solid molecule comes and binds to it, so let's say that this is the solid molecule binding to it. So now it is going to undergo some conformational change, which is reversible, just like enzymes. Okay, so this reversible conformational change, which is going to happen in that uh, in th in this transporter. And after this conformational change, the solute will slip in, okay, and the transporter will come back to its original form, okay. This is just a layman way of drawing the schematics, so just pardon that. So this middle step, the way that enzyme, when it attaches to substrate, it forms enzyme substrate complex, and then it converts the substrate, oh, well, it converts the substrate. So let's say that this is the substrate to which enzymes goes and binds. So then it forms enzyme substrate complex, and then the substrate is converted into the product, right? So this is the way enzymes work. So this is an enzymatic reaction. So in the same way, the transporters, they when they bind to the solute, they form, they come into the occluded state. So this occluded state is it is similar to the enzyme substrate complex. So this is an intermediate step where the conformational change is occurring and solute is bound to the transporter, and that is why they resemble to the enzyme to the enzymes, and that is why we also call them permeases. And well, this enzyme like manner, this reversibility, due to this reversibility, this chemical reversibility is also mirrored into the physical reversibility. So that is why the inverted repeats are the ones which are going to help in the they are found, inverted repeats are found, so that the transporter proteins are formed. We'll discuss about this physical and chemical mirroring in some other lecture. Right now, the point I wanted to convey is that transporters are the ones where the physical binding of solute brings in the conformational change in the protein, while the channels are simply the pores. Okay, so these are those are the proteins which are going to form channels or the pores in the plasma membrane, so that the diffusion can occur, so that from the higher concentration of the molecule, that molecule can move to its towards its lower concentration. So I hope the diffusion, the simple diffusion and facilitated diffusion is quite clear to you all. So facilitated diffusion is saturable, unlike the diffusion, because in diffusion, so depending upon concentration gradient, molecules are going to move. So once the concentration becomes equal in the both side of the plasma membrane, the diffusion will stop. And this is same case in facilitated diffusion, but it depends on the number of transporters that are present on the plasma membrane. Let's say that there are two plasma membranes. In the first plasma membrane, there are 10 transporter presents. In the second plasma membrane, there are 5 transporter present, right? So the more is number of transporter, the more will be the rate of diffusion, because they need those transporter proteins to help in the movement, right? But in the diffusion, it is not the case, and that is why simple diffusion is non-saturable. Means the rate of the diffusion, it has linear dependency on the concentration. So the graph will look like this. So as we increase the concentration gradient, let's say that this is the concentration gradient, and here I'm going to, okay, so let me just change it. So here, let's say that on the y-axis I'm plotting rate, and on the x-axis I'm plotting concentration gradient. So as I increase the concentration gradient, so as soon as I increase the concentration gradient, the rate of simple diffusion is going to increase. So simple diffusion has the linear dependency on the concentration gradient, but unlike the simple diffusion, facilitated diffusion not just only depends on the concentration gradient, but it also depends on the availability of transporters. So more are the transporter, more will be the rate of the facilitated diffusion. So that is why, unlike the simple diffusion, facilitated diffusion is saturable. Means if the all transporter are busy with the solid molecules, so that is going to limit the rate of the diffusion. Okay, I hope that my point is clear to you all. So this the simple diffusion, it simply follows the law of diffusion. Fixed law of diffusion. Well, we are not going to discuss about kinetics here, but I just wanted to point this out, that the simple diffusion follows the simple kinetic laws, which is directly, or which is linearly dependent on the concentration gradient. So those are the two different types of passive transport, but in those two types, the movement of molecules which are happening across the membrane is the solid molecules. Okay, so only solid, whenever the solid molecules move across the plasma membrane, it is called diffusion, and depending on whether it needs protein or not, it is categorized into simple or facilitated diffusion. But sometimes solvent molecules, they also move from one side of plasma membrane to the other side of plasma membrane, and that type of passive transport is known as osmosis. Okay, so let me just write it somewhere here. So the third type of passive transport is osmosis. Okay, so osmosis again does not require any kind of energy, any form of energy. So across the semi-permeable membrane, so let's say that here is the extracellular matrix and here is the cytoplasm. Okay, so let's say that this number of solid molecules here is more, and number of solute molecules here is less. So this is the concentration of solute. Okay, so solute concentration is higher on extracellular matrix, and solute concentration is low on the cytosolic part. So when the diffusion of the solvent molecule happens from the from the low solid potential to the higher solid potential, that is what we refer as the osmosis. So let's say that here the whatever solvent is, it is water. So in the water, solute is dissolved. So and this is a semi-permeable membrane. Okay, so this is very important point, that across the movement of water molecule from the lower solute concentration to the higher solid concentration across the semi-permeable membrane is known as osmosis. So water molecules will move in this direction from here to here, from A side to B side, and this movement of water molecules is known as osmosis. So osmosis is movement of solvent molecules. So solvent could be water in this case, or some non-polar solvent. Okay, so unlike diffusion, osmosis is movement of solvent molecules. Diffusion is movement of solid molecules. So please keep remember this important point. Then other type of passive transport that is seen in cell is known as filtration. Okay, this is usually seen in the kidney cells, the glomerulus. Okay, you have learned about this in your high school level. So unlike diffusion or osmosis, where the type of molecules is fixed, whether the solute is moving or solvent is moving, that is fixed. In this filtration, we define filtration as the movement of either solvent molecules or solid molecules across the membrane due to the hydrostatic pressure. So in diffusion or in osmosis, the movement was dependent on the concentration gradient, but in the filtration, movement depends on the hydrostatic pressure. Okay, so molecules, they are going to move from the higher pressure to lower pressure. So wherever the hydrostatic pressure is low, they will be moving towards that, and this movement occurs through the permeable membrane. Okay, so through the permeable membrane, if the water molecules, if let's say water molecules, means the solvent molecules are moving, or it could be the movement of solid molecules, but that is dependent on the hydrostatic pressure. So that does not depend on what is the concentration of solute in across the plasma membrane. It only depends on the hydrostatic pressure. So this is usually seen in the kidneys, in the glomerular filtration, where the small solid molecules and water, they could pass, and the large protein molecules, they are retained. So this is just like the sieving effect. Whenever we make tea, we sieve it through a membrane. So that is what we call it as filtration, right? So filtration has the same literal meaning in the context of cells, but the movement of either solid or solvent molecule, that depends on the hydrostatic pressure. So these are the different types of passive transport that could be seen across the membrane. Okay, so let's just quickly summarize all of those different types of passive transport. So here I have a summarized version of all different types of transport that we are going to discuss in this lecture. So those first four, these are the passive transport types, and the latter ones are the active transport. So we haven't discussed about active transport yet, but before that, let's just summarize the whole passive transport, different types of transport that we learned. So depending on whether which kind of molecules are moving across the plasma membrane, we are categorizing them into diffusion, osmosis, and filtration. Okay, so diffusion is movement of solute molecules, osmosis is movement of solvent molecules, and filtration is movement of either solute or solvent. Right? Clear? The diffusion, whether the diffusion is aided or not, whether the solid molecules need help to move across the membrane, depending on that, it is categorized into simple diffusion and facilitated diffusion. In the simple diffusion, there are no proteins involved, means this is unaided version of the movement of solid molecules. While if you need the help of the proteins, such as channel proteins or carrier proteins, which are also known as transporters, that type of diffusion is known as facilitated diffusion. So both of the diffusion categories, the diffusion is dependent on the concentration gradient, and the direction is downhill, means from the higher concentration towards the lower concentration, solid molecules show the movement. The examples are the gases that move across the plasma membrane, and the glut transporters that are present on the membrane of the neurons, that help in the movement of the neurotransmitters, that are also present on the glial cells. We have learned this about we have learned about this in detail in the neurobiology, right? And the aquaporins, the channel proteins that are going to transport the water molecules. Okay, the simple diffusion is non-specific, means any type of molecule depending on the concentration gradient it can move. But facilitated diffusion is specific. Why? Because the proteins are involved. The proteins that are going to facilitate the movement of the solid molecule, that are specific, means let's say that for example, aquaporins. So this channel proteins, they only okay, they only show the movement, they only let the water molecules pass through them. So they do not let sodium ions, they do not let chloride ions or any kind of other molecule to pass through them, for example gases. So due to the presence of the protein, due to the help of the protein in the facilitated diffusion, this type of diffusion is specific. While the simple diffusion is non-specific, you just need the concentration gradient, you just need the simple concentration gradient as the motivation, so that the any type of solid molecule can pass across the plasma membrane. And that is why the dependence, this is simply dependent on the concentration gradient, and that is why it shows linear rate. While unlike the simple diffusion, facilitated diffusion is saturable. Okay, so so this is about the movement of solute molecules. So if the movement of solvent molecules occur, such as water, that is what we refer as osmosis. Again, it does not require energy because it is a type of passive transport. The proteins that are involved are the aquaporins. So the direction of the movement of the water molecules or solvent molecules is from the low solid potential towards the high solid potential, which is also referred as the water down potential. Means the water is going to move from its higher potential to lower potential. So wherever water is at high potential, it is going to move from its high potential towards its low potential. So let's say in a solution, water is at its higher potential. So if the solvent molecule has the higher potential, means the solute molecules are at its low potential. So it is usually defined as the movement of solvent from the lower solute potential towards the higher solute potential, or in the direction of the water down potential. I hope it's clear to you all. The example is AQP2, and again, so osmosis is, it is affected by the solid concentration. So just to just for the your so just so that it is clear to you all, that this movement is just like any other movement, but the difference is that only solvent molecules are moving, and the the type of movement is affected by the solute concentration. So wherever, if in a solution you are increasing the number of solutes, so now the solvent molecules, so water is going to move towards that solution. So let's say that this is cell and this is extracellular matrix. Okay, so this is cell. So let's say that due to the certain metabolisms, due to certain metabolic activities, the number of solid molecules increased in cell, and those solid molecules are not being transported outside the cell. So there is no efflux of solid molecules from the cell. And let's say that the natural concentration of solute molecules is lower in the extracellular matrix. So due to the metabolic activity, the solute concentration is increasing inside the cell. So now the due to the increase in the solid concentration, now the water will move from outside to inside if this membrane has aquaporins. So let's say that this membrane is permeable to water, it has aquaporins. So now water is going to move from outside to the inside cell. So this movement, osmosis, is affected by the solute concentration. So wherever there is high solute potential, the water is going to move towards that direction. Okay, so this does not depend upon any other factor than the solid concentration. Well, it does depend on the presence of aquaporins. Okay. So the fourth type is known as filtration, which does not depend on the concentration gradient. It depends on hydrostatic pressure. So the pores are formed in the membrane, which give the sieving effect. So this is bulk flow from wherever due to the hydrostatic pressure. So this is usually seen in the kidney. The glomerulus filters the our blood, and this is based on the size. So like for example, small molecules, they will pass through the membrane and the large proteins are retained. So this is simple sieving effect that we usually see in our day-to-day life, and that happens same in our cells as well. So those are the different types of passive transport that we can observe in the cell. So let's now discuss about the types of the active transport.